Good morning and warm welcome from Indonesia to our honorable speaker and moderator of today's international guest lecture. Lecturers, students, and participants, before we start the opening ceremony, please allow me to introduce myself. I'm Agnes Anandita as today's Master of Ceremony. We all are here today with the spirit of the 61st anniversary of Fakultas Farmasi Universitas Pancasila. Today is such a great opportunity for us to have another series of international guest lectures on the topic of zebrafish laboratory and its applications in drug discovery. Ladies and gentlemen, before we move to the next agenda, we invite you to sing the national anthem of Indonesia, Indonesia Raya, followed by the hymn of Universitas Pancasila. For all participants, please be seated properly. Thank you. Ladies and gentlemen, the first agenda is the opening remark, which will be delivered by the Dean of the Fakulty of Farmasi Universitas Pancasila, Professor Dr. Apoteker Syamsuddin, Master of Biomed.
Thank you, Agnes. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Associate Professor Dr. Teo Seong Lim as speaker. The Honorable Lecturer, student, and participants. Especially as Dean of Faculty of Farmasi Universitas Pancasila, I express my high appreciation for the presence of Associate Professor Dr. Teo Seong Lim from the Faculty of Medicine, University Kebangsaan Malaysia, a speaker at the guest lecture. The topic of this activity is zebrafish laboratory and its application in drug discovery, which is important in providing broader insight and knowledge for students, especially increasing their knowledge of zebrafish laboratory. As we all know, zebrafish embryos are a very versatile model. They have fast development, and treatment can be easily performed simply by dissolving the drug in the water. Because of these characteristics, zebrafish embryos might provide important opportunities to accelerate the process of new drug screening. So, the topic of zebrafish laboratory and its application of new drug screening is important for us to know. This guest lecture activity is one of a series of activities in the framework of the 61st Faculty of Farmasi Universitas Pancasila anniversary. I hope that the present and all participants can take advantage of this moment to continue to gain knowledge from the speaker. Finally, we hope that this guest lecture can provide new insight, inspiration, and deep understanding for all of us. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Syamsuddin, for that wonderful remark. We are on to the next agenda, which is the photo session. So for all participants, please turn on your camera and prepare your best smile. All right, let's begin on the first slide. On the count of 3, 1, 2, 3. Okay, now we move to the next slide. On the count on 3. 1, 2, 3. And on to the last slide. On the count of 3. 1, 2, 3. Okay, thank you. Now let's move to today's main event, which will discuss further about the zebrafish laboratory and its application in drug discovery. Zebrafish offers a powerful instrument for researchers, facilitating the identification of novel therapeutic compounds, the repurposing of existing medications, and also the development of entirely new drug classes. This remarkable versatility stems from the zebrafish's ability to mimic human disease characteristics and respond to small molecules and drug treatments at physiologically relevant dose ranges. Before we begin the lecture, please allow me to introduce our moderator, Apoteker M. Iqbal Julian, RPP, PhD. Mr. Iqbal is currently a lecturer at the Fakulty of Farmasi Universitas Pancasila. He has several training experiences, including basic ethical review and good clinical practice. He is also an active researcher. Some of his publications are an investigation of the anti-hyperuricemic effect on Ganderosa leaves ethanolic extract in hyperuricemic rats and an investigation of biomarkers and handling strategy of erlotinib-induced skin rash in rats. Mr. Iqbal is already with us now. Good morning, Mr. Iqbal.
Good morning, Agnes. Nice to meet
Okay,
you.
nice to meet you too, Mr. Iqbal. So, without any further ado, the lecture session will be started. Mr. Iqbal, the time is yours.
Yes, thank you, Agnes. Good morning, lecturer, students, and all participants. As introduced by the Master of Ceremony, my name is Iqbal, and I am the moderator for the International Guest Lecture 7th Series. In this lecture, we will learn zebrafish, specifically the zebrafish laboratory and its application in drug discovery. The lecture itself will be presented by Associate Professor Dr. Theo Seong. Good morning, Dr. Theo.
Morning, Dr. Iqbal.
Okay, nice to meet you. Okay, first let me introduce our speaker. Associate Professor Dr. Theo is a lecturer in the Department of Anatomy, University Kebangsaan Malaysia, or UKM. He obtained his Bachelor of Biomedical Science as an honor degree and Master of Medical Science in Anatomy from UKM and conferred a PhD in Neuroscience from Monash University, Malaysia. His research interest falls in the neurodegeneration and neurodegeneration process in the brain of zebrafish. His work aims to decipher the differential neurodegeneration mechanism in the zebrafish in search of new treatment strategies for neurodegenerative diseases and brain injuries clinically. His current research focuses on using zebrafish as a model for Parkinson's disease. The exposure of dopaminergic neurotoxin, an example paraquat, rotenone, and MPTP, to the zebrafish result in loss of dopaminergic neurons, reduced locomotor activity, and dysregulated gene expression on dopamine biosynthesis. In 2021, he was awarded the Young Researcher Award from the Faculty of Medicine UKM and was listed in the top 2% scientists for single year impact, specifically in the year 2020 and 2023, released by Elsevier. Okay, that is our speaker introduction, and we can argue that Dr. Theo expert the topic of today's lecture. Before the presentation begins, these are the instructions of this lecture. The lecture will be presented for maximum 45 minutes. And after the lecture, the question and answer session will be open for 15 minutes. During the Q&A session, participants can ask questions directly to Dr. Theo or indirectly through the chat column. Okay, those are the instructions. Next, let us begin the main agenda of this event. Dr. Theo, if you are ready, you may begin your presentation.
Okay, thank you to the moderator, Dr. Iqbal. So let me just share my screen. Okay, again, so sorry for the trouble. Okay. Okay. Uh, okay, you all can see my screen clearly, right?
Yes.
Okay. Okay, so thank you. Good morning, everyone. So, first of all, thank you for thank you to Dr. Iqbal and also Prof. Samsudin for the invitation. And and of course, thank you to Universitas Pancasila for the invitation for me to give a short talk on zebrafish. Now, so I am Dr. Teo from Anatomy Department, Faculty of Medicine, University Kebangsaan Malaysia, in Malaysia. So my topic is here, like I'm going to talk about zebrafish laboratories and how to use zebrafish in drug discoveries. And I will share a bit more specifically on brain pathology because that's my research interest in neuroregeneration and neurodegeneration. So let us proceed to the slide. Okay, so just as a very simple introductions, right, on different models that we have used in biomedical research. We have used a lot of different models, and each of these models has important role to advance biomedical research and enhancing our knowledge in the pathogenesis of different human diseases, as well as different discovering new treatment modalities in these human diseases. So let's move on to the, let's say, the simplest model, which is cell culture. So cell culture is easy, as in, but it has its limitations, uh, especially when it comes to neuroscience, where if you use neuron cells, cell lines, or primary cells, the main limitations of cell culture is that the survival of these cells is dependent upon the culture condition. And especially in neurons, the cell lines doesn't develop the natural network. In another word is that the cell lines doesn't have the normal physiological afferent and efferent connections. So that's the main limitations in terms of cell culture in biomedical research, especially on neuroscience studies. Then next, we move on to a more complex model, which is the invertebrate models that we have the fruit fly, and we have the nematode, which is the C. elegans. Now, both models has been used for many years as medical models. This invertebrate model has a lot of similar basic cellular process. They have the basic gene functions as in humans or in mammals. And they are not cheap. I mean they are cheap. They are not expensive. So they are economical multicellular organisms that we can use, especially in large-scale experiments. And of course, the major drawback of these invertebrate models is that they are not mammals. So they do not have the similar brain structures or some organ systems like the humans. Therefore, they may not be able to model human disease accurately. Then we move on to another different types of model, which is the vertebrate models. We have the rodents, we have the non-humans primates. So these vertebrate models are highly conserved and very similar to humans. Especially when we talk about rodent models, mice and rats, these are the traditional animal models for us to study human diseases. These rodents, they are also mammals, so they have relatively similar anatomy and physiology to humans. And in the case of mice, we have a lot of transgenic animals that has been developed. However, uh, despite their small size, mice are still considered very expensive to keep in a large numbers. And the transgenic animal models that we have, they are very labor intensive to actually produce and to keep. So in this study and in this presentation, I would like to share something, another alternative models that you can use in biomedical research. That's the zebrafish. This zebrafish has been widely established as a medical model, especially in recent years, and this zebrafish has the combination of advantages of the vertebrate models as well as the invertebrate and cell culture models. Now, in the next few slides, I will share more about all of these advantages of using zebrafish in biomedical research. Now, just a bit on introductions on zebrafish. The scientific name of zebrafish is Biru. So zebrafish is a small tropical freshwater fish. They are present in rivers of India, northern India, in Bangladesh, in Nepal, and these zebrafish, they are active during the day and sleep at night. So the circadian system rhythm is the same as in humans. And the size, they are very small. In adult, it ranges between 2.5 to 4 centimeter long. The lifespan in lab, it can go up to a maximum of 5 years, but usually between 3 to 5 years. And in its natural habitats, it's recorded to be around 2 to 3 years. And here, just a bit of data information. Here, the numbers of publications per year that you can find in PubMed Central has been in the trend of increasing. So as compared to last year, in 2020, it's around 13,000, and in 2023, which is last year, there's a total of 15,000 publications in the year alone, in 2023 alone. So the trend is increasing. So because of obvious reasons, why zebrafish is gaining popularity. So let's talk a bit on that. So zebrafish here, this I think it has been mentioned by the MC and also Prof. Kamsudin that the zebrafish, they have transparent eggs and larvae. They can breed a lot. In a pair, they can breed around 100 and 200 eggs in one day. And the zebrafish, even in adults, is very small in size. They develop rapidly. So these are some of the advantages of using zebrafish. And it has the advantages of cell cultures as well, being low cost, being small, which means that they can actually run a lot of high throughput assays, as in toxicity tests or in drug identifications. Zebrafish can allow us to use, especially if you want to use the embryo or the larvae, you can do a lot of high throughput assays. And it's easily dosed. You can easily expose zebrafish to the different treatments that you use. And then moving on, it's a vertebrate, similar to rodent. It is similar to rodent. It is a vertebrate, although not a mammal. So they share high genetic homology to humans as compared to invertebrate models. And they have almost all similar organs as in rodents, except for the respiratory systems. And there's another point which is very important. That is this one. According to the UK Animal and Scientific Procedure Act 1986, they have listed that zebrafish, which is less than 5 days post-fertilizations, are not considered a protected species because at this point of time, the larvae is unaware of pain. And because of this reason, it is much more ethically acceptable to use large amount of embryo and also larvae to perform high throughput assays for preclinical studies for human diseases or drug development. So here I'm going to clearly mention, elaborate what are the other advantages of using zebrafish as why zebrafish is popular in using as biomedical models. The first one is because of the high genetic homology to humans. Zebrafish shares around 70% of their genome with humans. More than 80% of these disease proteins are conserved between humans and also zebrafish. Having this high similarity means that the drugs that are active in humans usually have the same target in zebrafish. So zebrafish, and so this is one of the very important point of like when we want to relate our findings in zebrafish, how can it be translated to humans? And this is another diagram just to show you. Let's focus on this gene MT2 here. MT2 is the metallothionein 2. I showed the gene, neighboring genes as well in humans, comparing to mouse and also comparing to zebrafish. You can see all the neighboring genes are the same. All the neighboring genes are the same for zebrafish and the humans, and also in rodents. Now, except for this part, that the TRADD genes and also XOST6 genes, they are translocated to another chromosome. But generally, you can say that even not just the particular genes, but even their neighboring genes are the same in relation, in arrangement, when compared to humans and also mouse. Then another thing is that the zebrafish has similar structures in terms of central nervous systems. We have a lot of homologous brain structures compared to that of human beings. We have identical cellular and also synaptic structures comparing to humans. Zebrafish also has the same arrangements of brain, that it has the telencephalon here, you have the diencephalon here, and this is the rhombencephalon, this is the cerebellum, which is all found in the in the rodent as well as in humans. Also, zebrafish has identical neurotransmitter networks like in mammals. You have the dopaminergic systems, you have the serotonergic systems, as well as the cholinergic systems. Now, this is one of the reasons why zebrafish is important in biomedical research model. Because zebrafish embryo and larvae are transparent. They are developed outside the uterus. At the same time, every pair of breeding pair can generate 100 to 200 embryos in one day. So because the unique transparency of the embryo as well as the egg, as well as the larvae, this allows us to study the details of development, starting from fertilization until they hatch through live imaging. And so they develop fast as well. Their development is very fast. Like desperation begins around 6 hours post-fertilization. All major organs will form within day 1 of post-fertilization. The embryo will hatch around end of day 2 or beginning of day 3 post-fertilization. Now, because of this, it is possible to obtain a large amount of larvae and adults in a short amount of time. If you want to work on adult zebrafish, it's very fast for you to gain the amount that you need for your experiments. So now that's about the characteristic features of zebrafish. Okay, now I'm going to more on research part. I'll discuss about what I did using zebrafish. These are the two main research interests that I have. I'm working on neurodegenerative disease, in particular Parkinson's disease, and also I did some work on traumatic brain injury as well. And then I would also describe a bit on different models like the stroke models and how we can use zebrafish for drug discovery. Whether is it for toxicity testing or drug screening, I will describe a bit on that. So moving on, Parkinson's disease. Parkinson's disease can be modeled in zebrafish. You can either use transgenic model or you can use neurotoxin model. So in my lab, I use neurotoxin models. And these neurotoxins that I use, they are known to be attack generally the dopaminergic neurons. Like for example, the toxin that kills dopaminergic neurons specifically, which is MPGP. So this is one of the most commonly used toxins to kill dopamine neurons. MPTP, the active form of MPTP, which is MPP plus, they are selectively taken up by the dopaminergic neurons via the dopamine transporters. This will lead to inhibitions of mitochondrial complex 1 and then resulted in free radical formations, reduced ATP synthesis, and then ultimately cell death. Other than that, we can also use different environmental toxins, for example, especially pesticides, which are known to be associated with increased risk of Parkinson's disease. One of it is rotenone. It's also a mitochondrial complex 1 inhibitor, which eventually leads to neuronal death. Another one would be paraquat. Paraquat, it's another pesticide that is known to increase risk of developing Parkinson's disease. The structure is very similar to MPTP, but it is not a substrate of DAT, and at the same time, it is also not a complex 1 inhibitor. Paraquat acts as a redox cycler, which enhances oxidative stress in dopaminergic neurons, which eventually leads to neuronal death as well. Now, how do we expose the zebrafish to these neurotoxins? We have various ways to expose neurotoxins or other drugs to zebrafish. These are some of the examples. We can do intracranial injection. We can do water immersion. The drugs, or in this case, the rotenone, the neurotoxin that I use to induce Parkinson's disease, you can just put it under the water, submerge the fish in the water for some time. So another one would be intraperitoneal injections. So the intracranial injections, intraperitoneal injections, both of these are widely used in rodents as well. And then in the case of Parkinson's disease, some of the common parameters that we can do is either gene expression studies, protein expression studies. We can use the brain to examine the dopamine level. We can do immunohistochemistry to check the dopaminergic neurons. And we can also do behavioral studies, for example, locomotion in case of using novel tank tests. So some of the results here, like MPTP or these neurotoxins, causes exposure to neurotoxins on zebrafish using on this PD model basically shows that the brain has aggregations of synuclein, which is found in humans as well. Like here, this is the Western blot picture showing that this is the synuclein protein, that the MPTP with 2 doses show increased band here compared to the control. And in the case of rotenone here as well, this is the Western blot as well. So this is the alpha-synuclein, which is in small size, low molecular weight. So there's not much difference between the control and rotenone. There's nothing here. But if you look into the higher bands here, which is higher molecular weight, more than 250 kilodalton here, the rotenone group has a higher band, darker, denser band compared to the control. This is another diagram here showing the immunohistochemistry on gamma synuclein. where here this is the control group and this is the rotenone treated group, that you have more immunopositive stainings compared to the control group, as you can see here. Then these PD models, they show dysregulations in genes related to dopamine action and also function. Like we check dopamine uptake, BAT, all of these reduce. And then when we go into biosynthesis of dopamine, which is thyroxine hydroxylase, this is the rate limiting enzyme to produce dopamine. We have a different pattern, as you can see here. Usually MPTP and also paraquat significantly reduce the expressions of TH1, but in the case of rotenone, it shoots up. Actually, TH1 expressions increases. We are not sure why the rotenone expressions increases the TH1, but when we look into the dopamine level in the brain, all of these groups actually decrease. Like MPTP is decreased, rotenone increase, but in the case of paraquat, it increases. One of the explanation is that because there is reduction of expressions of BAT that takes up the dopamine. With that, there's more dopamine that is found in the extracellular extracellular surface. That's why it increases the dopamine level in paraquat. Then this is the sagittal sections of the zebrafish brain. And these dots here represent the dopaminergic neurons. And we can divide the dopaminergic neurons according to their groups. Like, for example, this is the telencephalon group. Here we have the preoptic group. Here, these are the diencephalon groups and pretectal, and these are the rhombencephalon groups. So we can say that the PD models showed general reduction in terms of dopaminergic neurons, or when we compare to the control. So as you can see here, all the exposure of these neurotoxins reduces the numbers of dopaminergic neurons. Then when we move on to the behavioral studies. One of the easiest behavioral studies is the use of novel tank test. Here, you just put the fish into the tank, a new tank, and you record it for 5 to 10 minutes. And using behavioral software, you can actually identify the distance traveled for the fish for the duration of the 5 minutes or 10 minutes to check. And the neurotoxins exposure reduces the locomotor activity, as it's the same sign and symptoms that you can find in Parkinson's disease patients. Parkinson's disease also has a lot of non-motor symptoms like anxiety, dementia. Using the novel tank test also, we can check whether the fish anxiety level of the fish. Like here, we can just actually check the pattern of the fish travel. In a normal condition, when we expose the fish to a novel environment, the fish will stay at the bottom of the tank. But as time goes by, the fish will start to explore the upper half of the tank. So in this situation, the fish, in this behavior, we can conclude that the fish has decreased anxiety. So the more time that the fish spends on the lower part of the tank means that the fish are experiencing anxiety. And this is present in the neurotoxin group, where the fish spends more time in the lower half of the tank compared to the control group. Here, this also shows the marking trajectory marking of the fish. You can see that the fish has more time in the lower half of the tank. Then, we can further check different parameters. For example, PD models exhibited neuroinflammations. Here in paraquat and also the rotenone, the neurotoxin groups exhibit increased expressions to more or all the pro-inflammatory gene expressions, but decreases the neuroprotective BDNF, the brain-derived neurotrophic factors. And if you look into lipid peroxidation also, somehow the rotenone increases, but the paraquat reduces. Here, this part, we are still looking into why paraquat reduce lipid peroxidation. We have no answer to this yet. So that's about it about Parkinson disease model, and we do a lot of other studies, for example, microarray to identify the genes or the pathway that Parkinson disease, neurotoxin-induced Parkinson disease is involved, but I do not have the time to actually explain the results. So next, move on to another model, which is traumatic brain injury model. So traumatic brain injury model, they are a lot different types of models, and they are all established. And they basically have the similar models like in rodents. For example, using traumatic mechanical blow to the head. And in this case, we can just use a needle to perform a stab wound injury to the telencephalon. So this here, this is the puncture hole in the skull. And then if you expose the skull here, then you will be able to see this is the telencephalon and this is the injury site. This would be the injury site of the stab wound injury. And we can dissect the brain and then take out the brain to run numerous studies, numerous parameters. It's important to know these two date timeline when it comes to traumatic brain injury, depending on what you want to look at. At day 3, that's the peak of inflammatory response, and around day 21, that's the peak for neuroregeneration in zebrafish. So just a bit of results here. This is if you do a immunohistochemistry, this is the stab wound injury site. I'm looking at the expressions of metallothymine, which is a neuroprotectant. The cells here, the green fluorescence here, these are the NT2 mRNA expressing cells. So after injection, after the injury, the neuroprotectants are expressed more in the injured site compared to the uninjured site of the telencephalon. And then when we look into the proliferating cells here, this is in control. These are the proliferating cells. And of course, this is the injured area, so the proliferating cells are much more abundant in the injured lobe compared to the uninjured lobe here. And this is the treatment. Once we give the human metallothymine to another neuroprotectant, we can see that the numbers of proliferating cells increases. Then moving on, the apoptotic part, we can use tunnel assay, comparing to this is the control and this is the neuroprotected treated groups. So they are almost the same. So yep. So this is some of the research that I did in my lab. So then I will move on to stroke model. So stroke model, I'm not the expert. I didn't do stroke model in my lab. So these are from previous from other studies. So yep. So in stroke models, in rodents, it has been widely used in rodents. Like the most common model would be the occlusion of the middle cerebral artery. But this comes with some major constraints when we do the middle cerebral artery occlusion in rodents, like because it's very invasive. Therefore, it lacks the spontaneous effect in human conditions. And then it is you basically need to open up the skull to perform the MCA occlusion surgery. Now, because of this, the stroke model using MCA or collusion is you cannot easily access the pathogenesis of stroke in real time or in an intact living organism. So in this case, zebrafish can overcome it can overcome some of these limitations compared to MCA occlusion in rodent model. So when we talk about stroke model, we can generally divide it into the ischemic model and also the hemorrhagic models. And both of these has been used in zebrafish before using different stages like in adult or in larvae. So I'm just going through very quick about how other studies uses zebrafish in stroke experiments. Now, one of the most common model of ischemia in zebrafish is to use hypoxia. You can do this in adult, you can also do this in larvae. The advantage is that a large numbers of animals can be submerged in a low oxygen environment to create ischemic events in the brain. So in the ischemic model, you just need a glass, you just need a airtight, air-sealed, clear glass apparatus that contains some water. And this glass here, you will need 3 items. The first thing is an oximeter. This oximeter will allow you to monitor the oxygen level in the water. Then the second thing is you need an opening that has a tube connecting from the outside to the water here. Now this tube here has to be connected to a nitrogen cylinder. And then this will allow you to perfuse nitrogen into the water. And then this would kind of like reduce the oxygen level within the water here. So with the oximeter here, you'll be able to observe the oxygen level. And when the oxygen level drops until 1.5 to 1.7, this is the range that you can use to induce ischemic stroke in the zebrafish. And then the third opening here is just an opening that opens into the external environment. So with this, right, you just need to, once it reaches this level, the water reaches this level, low oxygen level, you can put the fish in and then observe them, observe the zebrafish. So usually in zebrafish, there will be 4 stages of hypoxia. So, yeah. So this is a video showing the different stage of hypoxia. The first stage here is that the fish will swim at the top, because at the top here, the oxygen level will be higher compared to the lower part. So the first stage is that the fish will stay on top. Then in the second stage, the fish will start to lose its posture because of low oxygen in the brain, as you can see here and here. As these conditions continues, it will move to the third stage where the fish will stop moving or very brief moving. But if you look closely, the fish is still breathing, where the opercula, which is the gill covers, move. And in the fourth stage, the fish dies. So also in this study, if you want to induce ischemic stroke model, you need to observe the fish until they reaches stage 3, and then move them to a new tank with normal oxygen. So this model is easy to produce, but one of the limitations of this model is that it has a very high rate of mortality. So meaning that you may need to produce a lot, you may need to use more animals to induce ischemic stroke. So under this ischemic stroke model, the hypoxia basically reduces mitochondrial metabolism in the brain. As you can see from here, this is TTC staining, where it stains the formazole, which is produced by mitochondrial dehydrogenase activities. So here in the hypoxia group, you can see that there's less pigmentation, meaning that there is reduced mitochondrial metabolism in the hypoxic group, hypoxic brain. And then hypoxia induces lipid peroxidation, as you can see in the MDA level. And it also increases the productions of ROS here in the brain. So the hypoxia also reduces total distance travel, as you can see from here. And also it also reduces the numbers of crossing to the upper half, meaning that the fish is in an anxiety state. Then, other than the ischemic stroke model, zebrafish, you can also use chemical to induce stroke. So chemically induced stroke is much more effective and much more reliable compared to the hypoxia by using low oxygen chamber. And this also can be introduced at the larval stage. And one of the chemicals that can use to induce can to form thrombosis is this ponatinib, which is a thyroxine kinase inhibitor. And it's a leukemic therapeutic drugs that has been withdrawn because in humans it shows that it increases the risk of thrombosis and stroke. So you can just inject a 1 microgram of per mil of ponatinib to a 2-day post-fertilization larvae. Now, the major drawback of this model is that you need micro-injection to a very small larvae, which means that it is very technical demanding in this case. But if you have injected properly into the larvae, you can see that there is increase of hematoma here, thrombosis here. And then if you do a HME here, you will be able to observe the thrombus in the brain of the zebrafish. So ponatinib causes impairment in blood flow in this cerebral angiogram here. There's reduced brain blood flow, and also there's blockage in the blood vessels. The locomotor activity is also reduced in the larvae. And then it also increase. This is the yolk sac. This is the eye of the fish larvae. And if you check here, the cell death is much more apparent. This is kind of assay. The cell death is much more apparent in the brain. Then also ponatinib increases cerebral inflammations, the gene expressions of these pro-inflammatory markers like interleukin-1 beta, 6, 10, TNF alpha, and then kappa B all increase significantly. And also the ROS production is also increased in the larvae. So although the ponatinib chemical induction is easy to use, but it's it's very like I mentioned, it's technical demanding. So in the case of hemorrhagic stroke, modeling the hemorrhagic stroke, I think is much more applicable to drug discovery process, where brain-specific bleeding occurs spontaneously in the larvae, and the hematoma can be easily visualized. Because the larvae is transparent, so you can basically just examine the hematoma using normal stereo microscopes. So in this case, you can use atorvastatin to induce hemorrhagic stroke. So this drug is traditionally prescribed to humans at risk of cardiovascular disease to lower the high density cholesterol. Right, so this one you just need to expose the embryo to this drug, and then after that you can transfer them after they hatch and observe the hematoma. So The drug induced causes intracerebral hemorrhage, as you can see from here. And then it also causes disruption in terms of the cerebral vascular structures and also the hematoma volume here. So these are some of the applications in terms of stroke model. Then this is the second last part of my presentations, a bit on zebrafish in terms of drug discovery. So the zebrafish, especially the embryo and larvae, because of their small size and transparency, we can perform high throughput in vivo screening. Whether you are using small molecules in drug screening or toxicity assessments, now the zebrafish embryo and larvae can be performed. So like you just need a pair of male and female zebrafish. You have the eggs, and then you can keep the eggs into 96 well plate. So each of these well contains a different drugs, different drug combinations, or different doses, concentrations of extracts, and then these drugs will be able to be absorbed into the zebrafish embryo. And from there, you can do genetic screening, you can do phenotypic analysis, as well as behavioral analysis in terms of the larvae. And with this, you will be able to identify novel drug leads, which are then you can use them for further processing through toxicity testing, absorption, distribution, metabolism, excretion studies, and then perform on other preclinical models, and then eventually going into clinical testings. So the main advantage is that you can examine thousands of embryos in a well plate. So this is the high throughput function of zebrafish embryo or larvae. So now this is just one of the study that actually show you the applications of using zebrafish embryo. So where this group of people by Sun, so they screen the chemicals, more than 5,000 chemicals, which comes from Microsoft's library, and also another library which contains another 3,500 compounds, and then they put it into this well plate, examine the effect of these compounds on the numbers of dopaminergic neurons. So they just need to check the microscopic observation, whether after exposure of these drugs, whether the dopaminergic neurons increase or decrease. So based on these 5,000 compounds, they are able to identify 19 chemicals that have changed the pattern of dopaminergic neurons following treatment. And 2 compounds out of these 19 affects the dopaminergic neurons without affecting the embryo development. Now, one of these is nerypholin. One of these is nerypholin, which is a cardiac glycoside family compound that is widely used for treating heart disorder. So they are able to find that exposure of this nerypholin increases the numbers of dopaminergic neurons. And therefore, they are thinking about how to use this nerypholin to actually treat dopamine, to increase the numbers of dopaminergic neurons in Parkinson's disease patients. I think they are still working on this in other preclinical studies. Now then, moving on, toxicity testings. Toxicity testings, I have been mentioning about it. So again, using species embryo, if you were to use toxicity testing using fish embryo, you can actually look for this Organization for Economic Cooperation and Development. They have come up with a guidelines to test chemicals. Like one of these are the few guidelines that they have mentioned. Like each concentrations you have to kind of like expose to 10 to 20 embryos. For each toxicity testings, you need at least 5 different concentrations, where the highest concentrations you should preferably result in 100% mortality, and the lowest concentration should preferably show no effect. And then with this, right, you can actually identify the LC50, NOEC, which is no observed effect concentration, or even LOEC, which is lowest observed effect concentration values. So using on 96 well plates. Then this is one of my last few slides. Okay, so in summary, advantages of zebrafish model is that they are small size, they are cheap. With the transparent embryo and larvae, it allows us to perform a lot of high throughput studies. The fish develop rapidly, and therefore you will be able to maintain a high number of fish in your lab, and it is easier to maintain compared to a rodent model. So easy experimental genetic manipulations. You can, like I'm show you, you can do intracranial injection, you can do intraperitoneal injection. You can also just expose the fish to the drugs in water. At the same time, you can do easily, because the fish is developed externally, so you can do genetic manipulation easily. For example, you can produce your genetic model using CRISPR-Cas9 injection easily, compared to generations of transgenic models in rodents. But zebrafish also comes with a lot of limitations. One of it is that the zebrafish is a poikilothermal animal that lives underwater. So in terms of thermal regulations and respiratory system, they are different. Like I mentioned in the earlier slides, that the zebrafish model in the lab, they can stay up until 3 to 5 years. And this means that they basically have a longer lifespan compared to rodents. And another some of this is difficulty to sample blood or urine. And if you want to expose the fish to herbal substances that needs organic solvent to dissolve, this is also one of the limitations. It will be difficult to dissolve, to expose the fish with organic solvent. So a bit more on now I'm going to talk a bit more on zebrafish laboratory, just a few slides here. Where to get more information on zebrafish? You can just go to Zebrafish Information Network. Under this website, you will be able to get a lot of information on the research in the genome, in the resource, where if you go to under the resource tab here, there's this zebrafish book here. Yeah, so zebrafish book here basically has all the information that you need to do your zebrafish lab or to do your zebrafish experiments. It gives you all the general protocol in terms of zebrafish care, how to breed, how to culture the embryo and also the larvae, what can you see from the microscopes, and then the other different methods here. And they also have all the recipes for the reagents that you require for your zebrafish. Then under the same website, if you go under resources, you can actually look for embryos. You can get internationally, you can get the embryos, which is cheaper compared if you want adult pair. So if you want adult pair, you can get it from them so that you can breed yourselves. or you can directly get embryos from them. So under the same website, you can actually ask for transgenic animals as well. So the price are all here, so you can have a look at yourself. And when we talk about zebrafish laboratories, there are a few factors that you need to consider. The first one is facility or infrastructures. You need space. Definitely, you need space. You need space for fish tanks. You need space for breeding areas, and you need quarantine tanks. And also, you need space for supporting equipments like microscope, dissecting microscopes. And in terms of zebrafish, it's very important is the water supply and water quality. You need a proper filtration systems. And if you want to save on water, you can purchase the recirculating aquaculture systems that allows you to recycle the water from the fish tanks. And another thing is the water temperature controls, water control, and lighting control. Then another thing is the equipment and supplies. Again, the same thing, the aquarium, the tanks. You need different tanks for breeding. You need different tanks for raising larvae and keeping the adults. You need microscopes, serial microscopes to examine your fish embryo or doing any intracranial injections or intraperitoneal injections. And usually, most of the time, the serial microscope will be in the same room as the fish tanks. So with that, you need a microscope cabinet because having the fish tank will make the whole lab, humidity of the lab will be very heavy, and high humidity will cause the microscope exposed to fungal infections, fungal contamination. So you need a microscope cabinet that can control the humidity. And then lastly, it's basically on personnels and also trainings. You need to have staff expertise in terms of how to do animal care, laboratory techniques, and also safety protocols. Implementations in terms of safety protocols for handling chemicals, equipments, and biological specimens of zebrafish. Now, the main items to be used is basically all of this. Yeah, if your facility has if your institutions have the money, I would suggest you to invest in zebrafish systems, automated zebrafish systems. These systems usually they are recirculating water, so the water keeps recirculating without any needing to change new waters very frequently, and their monitoring systems are all automated. They check their pH, they check for nitrate, ammonia levels, and if it's too high, they will alarm you so that you just need to flush out the water and replace it with new water. Of course, you can also set up your own zebrafish systems yourself. Right, you just need a bit of plumbing work, so it can be easily done. So yeah, just a bit of acknowledgements to the collaborators and also students and also grant providers for all my things that I've shown today. And these are the references, and that's all. Thank you very much for your time, and thank you again for the invitation. Thank you.
Okay, thank you for insightful presentation, Dr. Theo. Next, we move to the Q&A session. For the participant who want to ask questions directly, please click the raise hand button, then inform us your full name, followed by the questions. As for the participant who want to ask questions indirectly, please type your questions in the chat column. Okay, as for now, we still have no questions yet, Dr. Theo, but I have several questions.
Yes.
And actually, Universitas Pancasila is assessing possibility to build a zebrafish laboratory in order to comply with the ethical standard
Mhm.
in conducting biomedical research, especially for in vivo study. Okay, thank you for explanation. I am curious for the application of the zebrafish, especially in biomedical research, more specifically in dose conversion,
In?
drug dose conversion,
Uh-huh.
or drug dose calculations,
Yes.
because one of the method we can dissolve the drug in the chamber, in the fish chamber. How we can adjust the dose, uh, as for example, we in human using paracetamol 500 milligram per oral, and how can we adjust that dose to the zebrafish? That
Okay.
one I'm curious. And for the next questions, oh, okay, maybe later.
Okay. So when we talk about conversion, dose conversion in zebrafish, right? This is one of the difficult part actually in terms of using zebrafish. There are studies actually that come up with some formula or calculator that you can actually use that. But I think some of them, they are very particular in terms of the drugs that you use. I think, if not mistaken, there's one group of researcher that actually have published, I think just recently, that they have come up with a formula for cancer drugs. So I'm not sure how much you can actually use the cancer drug formula and then apply it to other drugs. So but there is some formulas that is actually out there that you may be able to use, but I'm not too sure about whether uh will it be applicable to what you will be doing, the drugs that you will be exposing. Yeah.
So it means the type of what drug will be used will be one factor should be considered when
Yes.
choosing this in vivo study. Okay, thank you, Dr. Theo. And I'm curious also about how you do the brain collection from the zebrafish. Uh, because the fish is very small.
Yes.
Maybe you can explain to me or to
Ah,
us.
okay. So it's basically the same as taking the brain out from rodents. We
Mhm.
anesthetize the fish with a high dose of drugs, and then we kill the fish with high dose of drugs, or you can uh, put the fish under ice water for around 10 to 5 minutes. Then we can basically, with that, we can kill the fish. And then after that, we just need to put it under the stereo microscope or dissecting microscope.
Nice.
Yeah, and then we can just take out the skull and then take out the brain. So the brain is like, I think it's around 5 mm long, 2 mm thick. So it's small, but it is possible. We can do that. We can do that.
Yeah, thank you, Dr. Taylor. We have one question from the chat column here from Miss Deola. She said, thank you for sharing your knowledge, Dr. Theo. How to preserve zebrafish? What is need to be done?
Okay.
So far, mammalian experimental animals have been used because mammals have a metabolism similar to humans.
Okay. So I'm not too sure about the first question, like how to preserve zebrafish, meaning.
Breeding, I think. I don't know. Maybe you can ask Miss Deola.
Yeah, yeah. If you can explain further, like the meaning of preserving the fish, as in uh keeping them in the lab, or preserving it in terms of histology?
Is in zebrafish in continuously zebrafish in water or like mungkin
Yes,
maybe
yes,
like that.
yes, yes, yes.
Yes.
The longest fish that I keep in my lab now, it's 2 years because I'm using them as a breeding pair. So and they are still alive. They're still alive.
Oh, okay.
So it's you can keep them for a long time. You can keep them for a long time.
Oh, okay.
Yep. And so as long as the water quality is okay, there's no problem. When we talk about the second part of the question, whether the zebrafish, how close is it for humans? I've explained it in my early slides. Genetically, 70% genome is similar to humans in zebrafish.
Oh.
70%. So I think, if not mistaken, rodent to humans is 80%. So fish to zebrafish to humans is 70%. So still a bit far compared to rodents, because rodent is still mammals, but it is much more better compared if you compare to invertebrate models. The main idea here is, I'm not telling other people to stop using rodent. Okay, but using zebrafish, it can complement your research.
Oh, okay.
Yeah,
Okay,
because
okay.
if you want to do
Okay.
high throughput study, you can't actually do it in rodent. Do it in rodent. So zebrafish is another method, another alternative models that
Alternative.
you can use.
Okay.
Yeah.
Okay. Okay.
Thank you for the questions.
Thank you. Yeah. Thank you very much, Prof.
Thank you, Ms. Deola and Dr. Theo. Is there any questions from the participants? You can raise your or push the raise hand button if you want to ask directly. Okay, as for now, we still waiting for the questions, Dr. Theo, and I would also like to ask regarding the zebrafish drug discovery. Is there any drug have been found during this zebrafish method?
Yeah. Actually, I think in just last month, there is actually a review article that actually give a very good summary of the amount of drugs that has been discovered in zebrafish and is now undergoing clinical trials. So I think all the drugs listed there, it ranges from different anti-cancer drugs, cardiovascular drugs. Yeah. And in fact, if not mistaken, some of the studies actually shows that when we talk about drug discovery, we always talk about preclinical studies. You need to involve a few different types of preclinical models before you can move on to clinical studies. I think in NHS or NIH, they actually come up with a guideline that using zebrafish drug screening, you can bypass the preclinical part.
So we can avoid using rats.
Yes.
Okay.
I think that there is a few criteria, but there's a few criteria that you need to meet before bypassing the other preclinical models.
Okay, I understood. Yeah, thank you for your reply, Dr. Theo. And I think we already approached the end of Q&A session. Okay. The end of Q&A session marked the end of this lecture. I, on behalf of all participants, would like to thank Dr. Theo for his willingness to deliver his knowledge on the zebrafish laboratory and its application in drug discovery. I, on behalf of the committee, would also like to thank all participants who were already active during the Q&A sessions. Dr. Theo, the committee has something to present to you as appreciation for your efforts. maybe the certificate K.
Okay.
And this is the certificate, the appreciative certificate for you, for your willingness to share your knowledge to all of us.
Yeah, thank you, thank you.
Okay, that's all for today's presentation. Once again, thanks to all of you. Good afternoon and best wish. Next, I return this event to the Master of Ceremony.
Okay, thank you so much to Associate Professor Dr. Theo Songlin, our amazing speaker, and also Mr. Iqbal, our amazing moderator. It was such a great and insightful discussion. Up next, we would like to present the certificates of appreciation to our amazing moderator, Mr. Iqbal. Okay, thank you, Mr. Iqbal, for guiding through our guest lecture today.
You are welcome, Agnes.
Okay, so this marks the end of our event. On behalf of the committees, we would like to express our sincere gratitude to our speaker, moderator, and all participants who joined us today for this international guest lecture. We apologize for any shortcomings that may have occurred during this event. Once again, thank you so much for your attention. I'm Agnes Andita as today's Master of Ceremony, signing off. Wassalamualaikum warahmatullahi wabarakatuh. Shalom, Om Santi Santi Om, Namo Buddhaya, Salam Kebajikan, Salam Pancasila.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you again. Thank you
You're
again.
welcome, Dr. Theo.